A recent study by researchers at the University of New Mexico has revealed alarming levels of microplastic accumulation in human brains. The study, which analyzed 91 brain samples, found that the concentrations of microplastics were 10 to 20 times higher in the brain than in liver and kidney samples from the same bodies. This suggests that the brain may be a primary site for microplastic accumulation, raising significant concerns about the potential neurological impacts of these pollutants. The discovery is particularly concerning given the results of a separate study, which found that 24 out of 30 blood clots analyzed also contained microplastics of various shapes and sizes. However, it's important to note that these findings have not yet been peer-reviewed. The presence of microplastics in the brain is troubling because these particles can cross the blood-brain barrier, potentially causing oxidative stress, inflammatory responses, and disruptions in brain function. Previous research has shown that microplastic exposure can lead to cognitive decline similar to dementia in mice, and there is growing concern about the potential links between microplastics and neurodegenerative diseases like Alzheimer's in humans. Although the full extent of these health impacts remains unclear, the accumulation of metabolic changes observed in animal studies over just 4 weeks suggests that long-term exposure could have serious consequences for human brain health. Human exposure to microplastics occurs through ingestion, inhalation, and skin contact. It's estimated that humans consume about 5 grams of microplastics per week, roughly the weight of a credit card. These particles can enter the body through contaminated food and water, as well as the air we breathe. Once inside the body, microplastics can cross the intestinal barrier and spread to vital organs, including the brain, liver, and kidneys. This widespread exposure highlights the pervasive nature of microplastic pollution and the significant challenges in avoiding these particles in everyday life. The increasing concentration of microplastics in human tissues reflects the growing problem of environmental plastic pollution, underscoring the urgent need for global action. Researchers are calling for more studies to fully understand the health implications of microplastics, as current research has primarily been conducted in laboratory settings or on animal models. Future research priorities include developing improved methods to detect and measure microplastics in human tissues, investigating how microplastics penetrate the brain and their long-term effects, examining the impact of chemical additives and plastics on human health, exploring the potential links between microplastics and neurodegenerative diseases. The potential health risks posed by microplastics demand immediate attention to protect future generations from their harmful effects.